Okay, so for the next half-ish, I'm going to go talk much sort of more closely about the work that sort of I've been involved with over the past 10 years, and particularly this question of dopamine in decision-making, and trying to use the neural basis of decision-making in and of itself is a very interesting question, but it's also, I think, a useful one here to unpick some of the questions about the role of dopamine in behavior. And again, I'm just going to sort of set up an extreme cartoon as a starting point, but just to sort of illustrate this question. So I guess one of the possibilities is that dopamine plays a role in economic decision-making. And so what I mean by that is, so this is a paper from Armin Lack, and again in Wolfram Schultz's lab. It's a nice paper actually, done relatively recently. So one of the things you might have been thinking from those first things, and when I was sort of saying, well, we don't know what reward means, is that obviously one of the things the brain's very clever at is ranking things in terms of prioritizing things for behavior. And what they looked at here was not just the different sizes of juice, so 1, 2, or large or medium or small drop of juice, but also they're going to add in small pieces of banana as well. And so for monkey B here, basically he would choose big juice over banana, or biggish banana, big banana over medium juice over small, and about equivalent for small banana and small juice reward. And quite intriguingly, there's certainly a number of the neurons seem to translate these different types of reward, one's liquid, one's food, into some kind of subjective value. So basically based on their choices. And then if you just present these in isolation, if you imagine that on average, you're going to get one of these, that when you get something over at this end, you should be positively surprised, and in this one, you'll be slightly disappointed. And what they found is that these saw exactly that kind of response, or the prediction error-like response. So a nice increase for the big juice, smaller one for the banana, and then decreases for the least preferred options. And then in a subsequent review article, Bill Stauffer from that lab described it here as dopamine cell activity codes reward value as a numeric, quantitative utility prediction error. So the idea with this is what we're saying is that we're going to transform current value based on current state of these into some kind of scale, which we can then use to drive our prediction error responses, and that these responses are appropriate for driving economic decisions. Yes?
Were all the cues for all these different rewards the same, or?
No, so each one was paired with a unique cue, so that the animals have learned cues associated with these. So what there is, though, is before any cue is presented, you think it could be any of these 5. So the average expectation is basically the average of these. And so therefore, when you get one that's this cue, it says it's a bit better than your average expectation, essentially. As I hope will be self-evident to lots of people here, there's also a very strong and interesting camp which has said, well, no, it's not really to do with driving decision-making or making choices between actions. It's really for motivating or activating the animal to respond. And obviously, this is something closely associated with Trevor, with people like Kent Berridge and Terry Robinson. And the example I've got here comes from John Salamone, who's done work over probably 30 or so years linking dopamine release, particularly in the core of the nucleus accumbens, with what he terms effort. So this is making repeated lever press responses in order to gain food. So this is a study where you've got a dopamine lesion of the core of the accumbens, a dopamine lesion of the shell of the accumbens and vehicles, and all the animal has to do is to press levers for reward. And what you see very clearly there is that there's a reduction in the amount of work that they're putting in to gain reward. And he's done much, this is just because it was the simplest way of displaying it. It's got some very nice things where essentially you can ask the animal, do you want to work hard for a slightly nicer reward, or just eat lab chow at the back? And what you find is this lesion doesn't change the amount of food they eat, but it's just that they become more indolent and will choose to eat the freely available reward rather than actually working to gain reward. And back in the 1970s, I think this is Sue Iverson's work, where if you make a dopamine lesion of the core of the accumbens and give amphetamine, you don't see that potentiation of locomotor behavior. So it's, this idea of dopamine and motivation and activation is very, very long in the literature and an important element. There's also, I think, a really interesting side that sometimes gets ignored about this, which there's a small community, and I think growing community, who are interested in dopamine and metabolic processing. So this is from a lovely sort of paper from Ivan de Rajal, who's at Yale. And all he did is he's using microdialysis in, this is in the ventral striatum. He actually thinks the dorsal striatum is perhaps more important, but this is ventral striatum. And he's either infusing glucose or L-serine directly into the gut. So this is not ingesting it normal ways. They've got a tube into the gut and they're measuring dopamine. And what you see is that there's an increase in dopamine in the ventral striatum when you've put something which gives you energy into the gut. And they've got a beautiful story about where exactly in the digestive system you place it. There's a certain point where you don't get ventral dopamine responses, but you do get dorsal. There's things about sweet taste and other things that come into this. There's a big story around this. But I just think it is really interesting that this is bypassing all of the normal things that we do, and you're getting this increase in dopamine. And I guess the tricky thing to try and think about how we bring these kind of ideas together is that they've all come predominantly from studies using a single technique. So obviously Wolfram, and also now, particularly now Achida's group, work exclusively looking at electrophysiological responses of these midbrain putative dopamine neurons, or identified dopamine neurons in terms of Now's case. Not always, but a lot of the motivation activation stuff doesn't come from recordings, but it comes from the effect of blocking transmission, often with pharmacological agents locally at the level of the terminals. And although, again, not exclusive, a lot of the metabolic processing stuff comes from studies of microdialysis. And even if it weren't, I always put this up because I think it's the worst drawing of a rodent in published literature. It's absolutely awful. I've got a similarly bad one that someone sent me of a monkey, which I can't find a way of ratcheting into talks unhappily, but I'm going to keep that for a long time. Okay, so what we've been thinking, and I realize in some ways I should have put this earlier, is that electrochemistry, and particularly fast-scan cyclic voltammetry, might provide one way to help us through sort of piecing these things together. Because you have the temporal resolution, which is closer to electrophysiology. I'll talk about it in a moment. But you're recording at the terminals, so you're actually recording release, and you've got a time scale which you can then really marry to aspects of behavior. So basically anyone who's come close to Paul Phillips will have some version of this slide. So if Paul had copyrighted this, he would be a rich, rich man. So this is our electrode. It isn't that big. It's 7 microns in diameter, which is important actually. So one of the things that Paul showed for the first time is that carbon fiber causes no gliosis in the brain around the tip. And so essentially you're recording from intact tissue. And that might sound, well, yeah, of course. But actually, if you look at the microdialysis probe after you've pulled it out, you're not recording from intact tissue in that case. You often cause some damage alongside. So I think it's an interesting, that's an interesting advantage of the electrochemistry. And what it relies on is the fact that chemical species that are around the electrode will oxidize and reduce a particular potential. So dopamine at a particular potential will oxidize. That will cause a loss of electrons as it turns to dopamine alpha-quinone, and when dopamine alpha-quinone reduces back to dopamine, you get a gain of electrons. And we can measure this change in electrons in terms of current at our electrode. So here's what we do. So in the case of fast-scan cyclic voltammetry, we hold the electrode at a negative potential, then we apply this ramp. It's actually not 10, it's 8.5 milliseconds. So very fast, where we ramp it up to a positive potential and back down. And we use that, and then at different potentials, different chemicals will oxidize and reduce. And so this is imagining it in a flow cell, so this is outside the brain, just imagining passing dopamine over it. And so what you'll have here is this is the rising part, the blue here of the waveform, that at a particular potential you'll be able to measure this change in current, and that this will happen at a very sort of particular position, and then it will just go back down, and then you'll get this reduction here as the dopamine orthoquinone gets changed back to dopamine. This is smaller than this here because you have some diffusion away from the electrode. You also, in the brain, you have some processes which basically work to oxidize dopamine orthoquinone, so it's not identical. And so if we take the peak of this response here, that has a linear relationship to the amount of dopamine in the vicinity of the electrode. And then if we take the shape here, so all I've done is I've switched it back on each other. So this is called a cyclic voltammogram, and essentially this is your chemical fingerprint that what we're looking at is dopamine. And in principle, you can dissociate lots of different chemicals. So here's that dopamine. It's flipped over because essentially that's correct. That's what you would see. We generally do the thing that people do in EEG, that we reverse it upwards just because it sort of makes more conceptual sense that this dip is actually caused by an increase in dopamine release, so we just invert everything. You can see here ascorbate. Serotonin looks similar. It's a peakier response. The reduction is positive rather than negative. So you can in principle distinguish these, and you certainly can in a flow cell. And then some metabolites. pH is an annoying one, which contaminates things, and we often, it's our biggest problem of trying to kind of get rid of when we're looking for dopamine. So we can actually, in principle, we can resolve dopamine from a lot of the other chemical species in the brain. The one major exception is noradrenaline, norepinephrine. So basically that has a pretty much identical cyclic voltammogram to dopamine. The good thing is when we're down, at least in most of the striatum, because the noradrenaline isn't in the striatum, we don't need to be worried about it. There's a slight issue in the shell, so that we can't definitely know that it's not noradrenaline that we're looking at. And obviously, if we're up in cortex, we call it catecholamine because we can't know definitively without doing extra experiments. But most of the time, we're fairly confident that we've got dopamine. And there's some really nice early experiments where they took TH knockout mice, or they knocked back in the noradrenaline, and if you absolutely hammer the striatum with a large electrical stimulation, you see no response whatsoever. So that really does suggest that what we pick up with voltammetry in the core of the accumbens is dopamine. So, and obviously what's good about it is that we can then basically look at it as animals do behavior. So you don't need to worry too much about what the animal's doing, but it's just that we can then really marry potentially trial by trial, moment by moment dopamine release with aspects of behavior. And so originally I sort of learned all of this and doing it in rats. I've become more convinced, and people in my lab thankfully do it, to work with mice as well to get access to some of the genetic tools. So we also do it in mice. And when it works well, and there's lots of times when it doesn't, you can get data like this. So again, don't worry about what's really happening because it's sort of irrelevant, but this is a 50-second snapshot of data from some task where basically a cue comes on. If the animal responds, then they get a reward. And this is relatively early with the animal learning it, so they're still sort of uncertain about things. And I just want you to sort of see that what you get here is, so basically the color scale here is basically the transformation of that current. So you're applying the waveform, going from negative to negative, that's displayed here, and times here going along this axis, and essentially that the color is the amount of current. And if you look along this line, that's roughly at the oxidation peak of dopamine. And what you can see is very clearly that you can pick out changes, transients to the cue, here to the reward. Here's a spontaneous one, not related to an external part of the task. And you can go along. And this is single trial resolution potentially. So you can really, when it works well, you can go back and look at every aspect of the animal's behavior and relate it to some aspect of the dopamine. As I say, it doesn't always look like this, but but it can do. The other really, really important thing to say about the technique, and definitely an issue with it, is that it's a relative measure. So this is not an absolute change. We're always looking at delta dopamine, the change relative to some baseline. We don't know what the absolute levels are. So in fact, 99.9% of the signal that you see comes from capacitance current, just from charging across the electrode that we get from applying that waveform, and you have to subtract that out. And so essentially these chemical changes, fractions of a nanoamp, are off the back of 1,000 nanoamp changes in capacitance current. So you're always looking relative to something, and that is an issue. So sometimes we don't know what those underlying tonic levels are, and that's obviously a potential. It's something that we have to be very cautious of, and means that it doesn't provide the whole story whenever we have this kind of data. Oh yeah, so, and obviously you can take at any snapshot to just to look across. So this is obviously effectively current by time using principal component analysis to get out a dopamine response. This is a current by voltage at a single time point, and there you've got your chemical fingerprint of dopamine. And again, this is a cartoon I've drawn for lectures rather than being fact. But if you sort of think about the types of techniques that we all might use, that you've got electrophysiology down here, beta, cellular, and temporal resolution, calcium imaging down here as well, and then sort of the human imaging techniques up here. Voltammetry sort of sits as not as good temporally or spatially as electrophysiology and calcium imaging, but what it really has is this chemical resolution. And I think that's its important part. And you can see compared to microdialysis, which has better chemical resolution, you're able to look at absolute levels, smaller changes, and multiple neurochemicals. It really sort of, what you get is advantages on the temporal and spatial scale. So, I told you in that first bit that dopamine release in the striatum, recorded with voltammetry, recorded with microdialysis, can look very different to cellular activity, the classic pictures of cellular activity. Keep that in mind, but put it to one side. Because what I'm going to do now is actually show you that when we're in the core of the accumbens, things look remarkably similar between the units and the voltammetric signal. So here's your classic learned cue response here. It's something I hadn't mentioned before, but it's important that obviously the size of the prediction error, the size of the cached value response, the learned value associated with cues scales as well. So as we get expectations of a larger reward from smaller reward, you see an increase in the size of the dopamine signal. And that's exactly what you see with voltammetry. So this is from Jeremy Day, working with Gina Corelli and Mark Whiteman, that using voltammetry, this is late on in training, so the animals have learnt the association between a cue and a reward, and you see very nicely a response to the cue and this tiny little response to the reward, but very little that's left there. This is from, I'll go into more detail about this study in a bit, this is from work that I did with Paul Phillips, where we're just looking at different reward sizes, and again, you can see a large reward is bigger than a small reward, which is bigger than nothing. So you get a good association between those two sets of data there. Again, something I'll come on to with more. One of the things that we've been very interested to do is think about different components of value. So obviously you might be motivated to do something by the amount you're going to get at the end of it, or you might be motivated by trying to avoid having to work very hard to get it. And whereas dopamine seems to care a lot about reward, we see a much less well-differentiated response to effort. And I'll come back to this, so just don't worry about that for the moment. But I think what's interesting is when groups have started to look at that with electrophysiology, see a very similar response. So this is from Ben Pascaro in Rob Turner's lab, that he's found that this is a regression coefficient. So this isn't actually the real data, this is how much variance in the data is explained by different factors in a reward-effort trade-off task. So reward, effort, the interaction, and other kind of things. And what you can see is this big, fast, positive influence of reward, as we've always seen. But here in green or the interaction in blue, there's very little negative influence of effort. And Chiara Varazzani and Sebastian Barret's lab has found a very similar thing as well. Prediction errors at the time of outcome. So sometimes when you're surprised. So here's a 50% Q, and again from Wolfram's data. And then here, they actually, on this 50% Q, this time they get the reward. So here you see this increase in firing rate at the time of reward delivery. Here, at the end of that Q, they didn't get the reward. And you see that dip in activity there. Not quite the same thing, but again, this is unpublished stuff from me and Mathieu Baudin in my lab, that we just did a switch where you either went from a low to high reward or high to low reward. Here, where you get a positive prediction error, you see this nice big increase, very extended compared to this, but that's again something that comes up quite often on the positive domain, and a little dip below baseline here in the negative domain. And interestingly enough, sort of no one's done this as far as I know yet with the units, but when we look for cost prediction errors, so basically the animals expecting to have to work hard and they work less hard, or working less hard and they suddenly work hard, we don't see anything in that domain as well. Even though the animals are sensitive to these parameters, they're changing their behavior, and we equate as closely as we can the value of moving from small to big reward as big to small effort. Okay, so I hope you can see sort of what you can get with voltammetry. I hope you can see what you can't do in terms of it's a relative measure, that there are these things. It's also, admittedly, it's tricky to do it. I mean, basically, we're getting fractional signals, and for reasons we don't quite understand, it works particularly well in the core of the accumbens, pretty well in the shell, quite hard to see signals in other brain regions, at least in rats. I think in mice, actually, again, we're finding that it just sort of the mice spurt dopamine wherever we put electrodes, but that's a different story. But it allows us, and it allows us to do things. And I think really importantly that although there is this heterogeneity, although there are things that can mean that release isn't the same as activity, et cetera, when you're in the core of the accumbens, what the cells do looks, the textbook story of what the cells do looks remarkably similar in lots of situations to the patterns of release we're seeing. So that's the starting point there. So then we can start to say, okay, I'm going to focus everything in on the core of the accumbens and ask what kind of parameters are encoded or reflected in patterns of dopamine release, and how do they relate to the decisions that are made. And the way that we went after this at first was the way that I guess probably a lot of people in the decision field were doing it, and had done it for a long time, which is to sort of take this type of model of a decision. So this is from Dale Lee and Minhua Zheng, but there's a lot of similar. I mean, there's lots of important things about this, and there's slight differences between models, but I think the detail doesn't matter for here. And essentially, the idea is that you have various options, whatever they are, and you're going to do some kind of value attribution. And again, I'm not going to talk about the complexities of that, but there's just some way that you're assigning value to these options. And let's say here that they're going to be associated with particular actions of choosing one thing over another thing over the third thing. So you've got value for here, from for here, from for here. You need to work out which one you actually want, and unless, so you have an action selection system. Again, don't worry too much about this. There's ways in which you might want to weight the way in which you select actions, so you don't simply always take the absolute most valuable thing. I think it was in Gary's talk, he talked a bit about the way in which the slope of the beta, the gain on some of these decision functions might change. Essentially, that's sort of built in here on the softmax function. You choose something, you do it, and you get an outcome. And from that outcome, you get this classic prediction error, the difference between your expectation up here and the outcome. And so what we were interested in is to try and sort of decompose the degree to which dopamine encodes something about these value functions. And again, to make our lives easy and to make your lives easier too, I'm going to just take situations where the world is relatively static. So ignore that bit and don't ignore it because it doesn't matter. It's important and matters a lot, and dopamine matters a lot for this. It's just trying to simplify the story. So everything I'm going to present you is situations where we've got reason to believe the animal knows the contingencies and is operating based on those known contingencies. Okay, so where we started, and basically this is work so I went to Paul Phillips's lab in Seattle, and essentially Paul had gone to Seattle, had this stellar postdoc where he'd really pushed forward voltammetry with behavior. I'd had a lot of experience in getting animals to do decision-making tasks and to be interested in this. We got talking and we realized there's a load of interesting questions we could do. We actually started talking, I think, in 2004. And yeah, so 6 years later, we finally got our first paper for when we actually managed to do it. The experiment we talked about on those first in 2004, I'll present second, that was published in 2014. So again, you can see that sometimes it takes a long time from great idea to conception, basically. So this was actually done sort of on the ground, particularly by Jiaolin Gan in Paul's lab. And the structure is sort of similar to the way that we always approach these kind of tasks. So we had our rat, we have an operant chamber, and with a food magazine in the middle, we have a lever on one side and a lever on the other. And the way that we often design these tasks is that we would give a certain number of trials where there's only one thing they could do. So they forced them to choose left, right, right, left here. And one of the things that was useful about this is we could then look at value-related dopamine in a very kind of isolated, easy manner, because we don't know, is he encoding this one and this one and this one and this one? No, we're just sort of getting something very sort of refined and focused. But we want to know what the animal actually how it subjectively values these things. So we'd also run mini blocks where they'd get a choice between the two. And so that gave us a behavioral measure of subjective preference. And again, in terms of what I'm going to talk about, I'm just going to talk about situations where they're choosing what we have determined is the high value option above 75% of trials. So that, again, we've got a behavioral evidence that they know the values of the options and they're performing in a way that makes sense to us. And obviously we can look at dopamine on these, and I'll do a little bit, but most of it comes from these forced trials. Okay, so this is the sort of task itself, and what we wanted to do is basically we had what we called a reference value. So one of the levers at any one point, they'd have to make 16 lever presses and then to get a single food pellet. And that was like their standard thing. And then what we did is we've manipulated the value of the other lever. So we could increase the benefit to 4 rewards, or we decreased it. And actually, this was a silly condition. This was, again, a good example of you do the better experiment after you finish. So we kind of realized that it would have been much more sensible to have 2 rewards to go to 1, but it's still an increase in a reduced benefit there. And then we also had conditions where they were going to get the same reward whatever they did, but here they're going to only have to put in 2 lever presses, so you've got a reduced cost, whereas here they're going to have to put in 32 lever presses to get that option, so they've got an increased cost. And what we did is, day by day, we reversed the value of this reference value. So the animals are always learning about where the good and the bad options are. I mean, if they'd done it enough, they could have sort of alternated and worked out the structure. We had no evidence that they did that. They nearly always started responding on what was the previously high value option on the previous day. And we could look at how they worked with this. And the important thing, again, I haven't got the slide to show it, we did an experiment where we said, actually, rats, would you prefer this one with increased benefit or the one with a reduced cost? So here you can either get 4 rewards at an FR16, or you can make 2 lever presses and get 1 reward. And we found that basically on average, the animals were at 50%. So that the subjective value of reducing the cost here is roughly equivalent to increasing the benefit. Okay, so you've actually seen this data already, so this should be sort of obvious to you. So in the reward conditions at the time of Q onset, so when they're presented with the options, that you see on these forced trials, you see this nice relationship with reward size. So 4 is bigger than 1, 1's bigger than 0. And so we always, because we had this reversal thing, we always ran it in 2 sessions, with one with high value on the left, one with high value on the right. And you can see it's very consistent across trials here, across sessions. But in those effort conditions, as I say, that's not at all what we found. And again, I should emphasize, this is where the animals are choosing the low effort option. So they've no evidence, they're just as quick to learn, and they're just as motivated to go to get the low effort option as they were to get the high reward option. And yet nonetheless, in most of the time, there's no difference between the dopamine release. So you may think, okay, well, so that's fine. So there's maybe nothing particularly about effort costs and dopamine, but there's a good relationship between reward dopamine and the decisions they're making. And that's true, but actually, I'm just going to show you evidence that I think it doesn't necessarily mean that one is really driving the other. So we had the occasional session where animals performed badly. So most of the time, so what you've got here is a behavioural performance of the animal from the sort of averaged from the start of the session through to the end of the session. So 0% from 0 to 1. And so they usually, as I was saying, start below 50% because they previously were preferring the option that is now bad. And over time, they'll learn, usually within about, I think it's about 20 or 30 trials, they'd reach criterion and they'd be performing up here. On about, I don't know, maybe sort of 5 sessions out of 80, if not fewer than that, you'd get performance a bit like this. So the animal improved a bit, but then sort of just stuck around at 50% performance. And looks like he's just got it at the end. Probably if he would run it for another 20, 30 trials, he would have come up and done that. Fine, that's okay, things happen like that. Chuck the data away, don't worry, do the averaging. Or you look at the data, and what we found was that even though the animal's performance was no use to us and seemed to be agnostic about whether to choose the left or the right lever, the dopamine knew which was the high value option from about trial 18. So red bar is the response on the high reward forced trial, or average of the pair. Blue is for the low reward. So you can see it's still got the previous session's value distinction here, so you can see that blue is higher than red to start with, but then by the third block it's switched. And every single block throughout that, even when the animal is performing at 50%, the dopamine is saying, that's the high reward, that's the low reward.
So do you alternate the forced trials and the choice trials?
Blocks of forced, 4 choice. And so yes, you might think, maybe there's something funny about forced trials. Maybe they're doing it differently somehow, and so that's why it's happening. This is the choice trials. Every time they choose high, you get high. Every time they choose low, they get low. So the dopamine knows what the animal should be doing. The dopamine knows that's going to give you high reward, that's going to give you low reward, but nonetheless, the animal for the whole session is not showing that preference. So I think that's intriguing. It's also almost impossible to know what to do with, because it's like this, basically you can't design an experiment to look at the occasional session where the animal does the wrong thing. So that's actually a book chapter, because that was the only place I could find to publish it. But we thought, okay, well, let's try and do an experiment to really bring this out. And this is the experiment that Paul and I originally designed back in, that's just to say dopamine preferentially encodes reward over effort, but the patterns of release are uncoupled from the choices that are made. So yeah, this is the experiment that Paul and I originally designed back in 2004. And this in some ways is the interesting experiment. This is where you ask the animal to trade off costs and benefits. So you're saying, are you willing to work harder for big reward, or you do want to take the small reward for less effort? And so this was done by Nick Holland, also in Paul's lab. And so it looks very similar, same rat, same box, same options. And what we've got now is, again, we sort of tried to control things a little bit. So we had certain conditions where we called them moderate cost conditions. So in this case, they're only going to have to put a little bit more effort in. I think they have to make 8 lever presses rather than 4 lever presses, but you'll still get a high reward for that. And the reason we wanted that is that we wanted they have to work harder, but we wanted them to be choosing this option most of the time. And then in a second condition, which we ran on different days, that we really ramped up the effort on the response cost here, so that actually the animals, on average now, preferred to choose the low reward option because it was easier to gain. So essentially we went up to a 32 or sometimes 48 or 64 lever presses. Again, I think probably some of the people here will know this from my previous talks. UK rats are a lot lazier than US rats. So when we piloted this in the UK in Oxford, we'd got very nice curves. And as soon as we went to Seattle, we kept having to ramp up this effort before we'd get them to switch. Okay, so this is the behavior. This just says that they did what we wanted them to. So here in the moderate cost condition, they choose the high reward at the medium effort, and they're faster to do that. But when we switch it, now that they've got a preference on average for the low reward option at the lower effort, and again, that you can see that they're faster to respond to the low effort, low reward, and the high effort, high reward. And you can see it's not that it's not scaled, so it's not that the low effort, low reward here is faster than it was here. So that relative scaling is really important. Yes?
So wouldn't a better way to look at this data be to look at to basically calculate the value of work as a product of effort and reward, and then see probably a very nice inverted U?
Yeah, I mean,
I
think yes, I mean, I think you're right, and we talked about sort of whether we should do it in a much more behavioral economic way, essentially. And I guess what we decided is in some ways we wanted to really hammer home the case that what does the dopamine look like when they're preferring a low reward option. And so I think you're right. I think the first version we had was something which is much more dynamic and tracking over time. This just, again, somewhat simplifies the story. So that's what the dopamine release looks like in the moderate cost condition, sort of as what you'd expect. So the high rewards, which they're preferred, there's a bigger response than the low reward. This is the key condition. So what do we see when you present the animals with a cue? So now they're preferring this low reward over the high reward because of the cost you've imposed. And what you see is there is a reduction in the peak response here. So there is a reduction, but it's no way is that lower than this. And actually, if you look out over time, if you were to take some kind of area under the curve measure here, as we did, that actually, I've lost, you can sort of mentally associate it. So that's 1 second of time. So about 5 seconds coming in at about here. You can see that essentially that this is a higher signal for the non-preferred high reward over the preferred low reward option. And actually, Nick went on and did a whole other experiment to show that if you presented the cue here but only presented the levers 5 seconds later, that you still saw this effect. So it's not just that they're working here at this point. Yes? Yeah, everything I'm talking about is accumbens score, and for everything from, yeah. No.
What happens if the rats have to choose between moderate and high cost?
Moderate and high cost.
Instead of choosing between low, and high medium, or low and high. Maybe because of the magnitude.
I think they would. So I think that's in some ways they would high. You mean would they choose high medium over high high?
Right.
Yeah. So they'd choose. They like to. I mean, in some ways they optimize. So they're going to go because you get the same reward here, but at less effort. It's a bit like our first study. Do you see what I mean? So that one, there was a lower, kind of lowish reward, but if we'd scaled everything up, we would have expected it. It's just that they're making the decision here on effort. So our assumption is once the animals knew the contingencies, that you'd see this overlap there in the dopamine because the animals are responding based on effort, not reward. And again, just to say this isn't to do with choice trials. I took out the one example, which I'll talk about later, where this falls down. But in all of the other stuff, it doesn't look like forced and choice make a difference. So these are the choice trials where the animal chooses high and chooses low, where both options are available, chooses high, chooses low, with both options available. Yeah?
If I got it correctly, only want to recap, and it's physical effort, because they have to press the lever, so do you find the same as mental effort?
We haven't looked, so I think it's a tricky experiment to do. I think someone sitting behind you tried to do this in humans, doing mental and physical effort. And it's a very hard thing to try and really equate, because when you do it in rodents, what you want is mental effort, but the same reward rate. And that's really hard to do because virtually everything you do to make the task harder, they also means their performance is worse. But I think it's an interesting question. Again, there's a whole interesting literature here, which I'm, again, going to skirt round because there isn't time, about what effort is. So John Salamone's has got the story that basically if you make the effort really just to press a lever hard once, you make the lever heavy, don't need accumbens dopamine for that. So what you need it for is this kind of this temporal extended use of effort. So we're working through time. And actually, I'll show you a little bit of data that maybe noradrenaline is exactly the opposite. So basically, noradrenaline you might need for the challenge of overcoming something hard work, but you don't necessarily, actually, I'm not going to show you this data, but you don't necessarily need it in a sequence task, basically. Yes?
Do they experience the high reward, high effort trials enough to learn about it?
Yeah, definitely. So we basically give them whole sessions of forced and choice, and so they're getting a huge amount. And so we're only running them once they've reached criterion of having a preference. Yeah.
So in the high reward, high effort condition, the difference between that and the other condition sort of emerges a bit later?
Yeah.
What is your interpretation of that?
Effort matters a bit, but just not a lot. I'll show you. I'll show you something which really kind of sums that up. There's definitely a difference, but what is never seems to, or like in a fraction of things session seems to happen, is that the red is underneath that. So it's somehow the preference sort of boosts the differential at that peak response, but what you never see is the flip. So if it was somehow really to do with your your drive and motivation to get this one, because reaction times are faster to these trials than these trials, then you ought to see the red below the grey-black there. Yes?
I know in Moscow that at high cost, some stress can start.
No, they like working. I mean, the 32, but 32 lever presses I don't think is so we never measured it, but my guess is that we don't see any sense that they don't mind doing this. I mean, it's not that they've got the option to do nothing, and they definitely don't. They work hard, and on progressive ratios or other things, they'll do 200 lever presses. They're hundreds and hundreds in the right circumstances. So I don't think it is that, certainly. So this gets at your question, Oliver. So one thing that Nick did very nicely, and I think was to try and come up with a way of displaying all this data. So he came up with a choice index, which is basically, do you prefer the high reward or the low reward? The more you prefer the high reward, you're over on the right. The more you prefer the low reward, you're over on the left. And then he did a very similar thing called a dopamine discriminability index. It's essentially looking at the way in which an ideal observer would discriminate the average signal in the high reward and the low reward. And then it's going to plot this all together. So again, just to sort of emphasize this, if it was a utility signal, we'd expect things in these quadrants. So if you chose the high reward and preferred it, then you'd get dopamine coding for it. But if you chose the low reward, then you'd see dopamine coding for the low reward. However, if it's really something to do more with reward coding, you're going to see everything up in these quadrants here. So dopamine coding for the high reward, irrespective of your preference. And that's all the data. So that's all the sessions from all the different manipulations and all the electrodes that went into this. And so what you can see kind of clearly is that there's very little down here. So there is a negative slope here. So basically that's the point you were making, that there is some effect of effort. It's not that it's completely irrelevant, but it's just that the main driver of this is reward size. And this is actually just to say, so this is not me, this is Sebastian Boré, so Chiara Vautani. So Seb did a really nice, painstaking study where he recorded in monkeys the dopamine neurons and local ceruleus neurons, and on a cost-benefit experiment. So what he found is, again, very similarly, that the dopamine neurons particularly care about reward and much less about effort. And then this is actually at the point where just before the action's made, if you time lock to that, the dopamine doesn't seem to care about what's coming up. There's sort of when you collapse across these at the time of action. Whereas if you look at locus ceruleus, you get a slightly different picture. So in LC, it seems to respond particularly to the large reward. And not really, I mean, there's some scaling there, but it's much less. But what LC cares about is, so this is the point of the action, And here we've got a positive relationship with the size of the effort. So for the hardest effort, effort 3 is the hardest effort, you get the biggest response after, as you're about to do and make the effortful response. And then that goes down for the lower effort responses. And what Sebastian Boré has been arguing, and I think it's a really nice idea, is that to some extent the roles here are that dopamine might be involved in the process of something to do with valuation of whether it's worth engaging with effort, i.e. what you're going to get in the end of it. And once that there's a decision come through about, yes, it is worth doing it, go for it, noradrenaline comes online to actually bodily prepare you, give you arousal to do that. And I think that's really nice example of what could be some distinction there between those two systems, even though both are intimately involved in effort-related decision-making. Okay, so basically, the end of this study, you can see the conclusion is identical, that dopamine preferentially encodes reward over effort, but release is uncoupled from choice. So that's sort of feeling relatively convincing. But I guess an interesting thing is that some people have argued, well, dopamine, the effort's a bit special, so other things, effort you can't really tie to an outcome. Effort's to do with action systems, it's hard to associate effort with cues, maybe that actually it's just something sort of particular about effort that decouples in this case, but this is like a one-off example, and that's, if we take another parameter space, everything's going to align better. So it's not going to, but so that's the conclusion, but I'm going to show you evidence of this. So this is work that was done in Oxford by Yorgos Papageorgiou. This is state-dependent reward. So this is, again, similar looking experiment. So two options here. There's subtle differences, but you don't need to worry too much about them. But essentially, the forced trials here is one of options associated with food reward. The other option is associated with sucrose solution. There were other things in this experiment, so we gave them some surprise where they sometimes got big rewards. We sometimes changed the identity. I'm not going to touch on that, but happy to talk about that later. And so we trained them in the baseline state, and we either devalued the food by pre-feeding for an hour or pre-drinking for an hour before the session, put them back in their baseline state, did a re-baseline, then gave them the opposite devaluation. And one of the things that sort of I think is quite important is that we ran this in a food-restricted state. So the standard kind of thing that our animals were restricted to about 85-90% of free feeding weight is very normal in these kind of studies. And one of the things we tried to do is, on average, have it that a bolus of sucrose was equal to one of their grain pellets. So you can see that this guy didn't think so and this guy didn't think so, but on average across the cohort that you're getting a 50% preference. So there's as much variance explained by animals that prefer sucrose as ones that prefer food. So this is just behavioral data. And importantly, it seemed to be very fixed within an animal. So here's pretest 1, pretest 2. This is baseline A, and that you can see that it's a very kind of fixed preference. And actually, interestingly, we ran the first cohort of about 2/3 of the animals, then put another third of the animals through, and we didn't have to change anything. So the parameters that we used for these first animals seemed to be so we seem to be capturing something about that a subjective similar value of sucrose bolus versus a food pellet here. And this is just saying that, so we do this devaluation session, which I'll show you in a bit, and that again, even after a devaluation session, you put them back in the baseline state, and again, the preference is really pretty stable here. So that's fine, that's a useful starting point. What's interesting then is that for some reason, the accumbens dopamine always preferred the food cues. So here, the response to the cue. Here we've actually, again, separated the cue and the lever extension, and you can see again, the dopamine response to the food cue is always higher than the sucrose cue. And even, it's very persistent, it's also here in the baseline session. So if we take that sort of thing that Nick Holland did of these kind of dopamine index versus a choice index, again, you see all the animals are clustered up in the top here. So irrespective of whether they prefer sucrose solution or prefer food, in those baseline sessions, the dopamine is larger to food in a food-restricted state than the sweet sucrose liquid in that case. Is that just something about food? Is that something funny? So I guess this was made the evaluation sessions important. So we could either, if we pre-feed, preference for food reduced. If we pre-drink, the preference for food increased. And then we reran the session. So we've also got the reinforcers here in the session. So it's not a learning thing. Yes.
I was thinking about the food restriction and food as something like object, and then sucrose in a liquid form. So, and what about like some water deprivation just a little bit before the...
Yeah, I mean, we couldn't do food and water restriction, but I mean, it's post hoc, but I agree. I think that I would like to link this to some of the metabolic type things, that actually calorically, a pellet of food is much better than a bolus of sucrose solution, even though the sucrose solution has, sweet taste and so might drive behavior for other reasons. But we don't know what those things are.
And also, sucrose is less nutrient than, for example, glucose.
Yeah, no. I think there are a series of interesting metabolic things that we could and are sort of gearing up slowly to think of tackling. Yeah, exactly. And there's really nice work from people like Jamie McCutcheon in the UK and others, and Mitch Reutemann again are actually really unpicking some of those things and the peptides that then interact with those. So that's, if you devalue sucrose, the sucrose response goes down, the food response stays high. Interestingly, you devalue food and you get the opposite. So the food response goes down. Actually, the sucrose response significantly increases. So again, I think there's something, so here obviously you can imagine if you've eaten dry food for an hour, you probably quite want some liquid, and that's the only case where you get an increase in the valued food, and the valued option post devaluation, whereas here that basically it's pretty static across conditions. Yes? No, they're grain pellets in this case. Grain, yeah, kind of cereal pellets basically. Okay, so again, there's lots of other things in that study and I'm not going to go into it, but I just want you to see that dopamine clearly encodes something, and I've called it here a state-based reward signal, and that's deliberately vague. I don't think we know what it is, but it's some interesting thing which reflects the animal's current state and motivation. But again, the important thing, that patterns of release is uncoupled from the choices the animal is making. So we've got 3 examples here. We've got, in the first study, the effort, they don't respond to effort, changes in effort in the dopamine, even though that behaviorally they do, plus the odd session. Here we've got the response to the reward over effort, even when they're subjectively preferring the low reward. Here we've got a kind of thing that just fell out accidentally of the data, that the dopamine response is greater to food than sucrose, and again, even though preference-wise we don't see anything about that. So that's odd, and I guess at some level that was sort of slightly worrying, because I'd written a whole 5-year program based on the role of dopamine in decision making, and at the moment what we were getting here is that the answer is there's no role in decision making. It's this epiphenomenal thing that dopamine's quietly shouting down in the accumbens about what's good in the world, and the animal's doing whatever it feels like. So I guess it's this and a number of things started us to wonder whether really we weren't looking at the right model of decision-making or the right parameters of decision-making. So the cleverer ones of you might say, that's not really an action selection model in reality, unless we assume that one of the actions there is none of them, inaction, don't do anything, leisure, as various people have called it. So there needs to be a motivation actually to go through this, and everything I've shown you is from trials they actually completed. Yes?
Just wondering if your action selection based on your softmax is stochastic, so random choice, but based on probabilities, how much you felt if it's automatically the highest probability that comes out of the
So I missed that last bit. So if the...
And with your softmax, you get probability for action 1, 2, and 3, which
Yes.
is sum to 1. So would you just automatically take the highest probability as your chosen action, or would it be like a stochastic choice with a preference for higher probabilities?
Yeah, so what we did with this is because we sort of set the parameters as there's clearly a right answer, like there's clearly a big reward and there's clearly a low effort, we just said, yeah, we're assuming that the softmax is pretty much a step function here, you know.
If your animal was building a trial of its action A, then if you had a stochastic choice, you'd actually get variability in the beginning and then converge towards the...
Yeah, so I think, as I say, I think when you go into dynamic environments, a lot of this might be more complicated, more interesting, and more linked together in some ways. And I think it's more that once you're in this steady state that a behavioural policy has been established, things uncouple, essentially.
So, and about the previous experiment, what would happen if the food was sugar-based? Because the taste was the same and the hormone releasing was the same, for example, the level of insulin. And just kind of the food and liquid was different. Not the taste it was from the...
So I don't know is the short answer. I mean, I think what we were trying to do is deliberately make them very distinct. So, because one of the things rats do quite readily is generalize. So we were sort of trying to make it really clear that if you did one thing, you got this outcome that shared very few of the properties of the other one. So I suspect that if you made them closer, I think A, they would prefer a sugar pellet. The same size sucrose bolus would be less preferred over a sugar pellet, so you'd have to increase the sucrose response.
Because now there are some different traits between these two kind of rewards.
Yeah, no, I think as was saying to someone else, I think that there's really interesting questions on the parameters of the reward and what elements of it are influencing the dopamine release, and I think that'd be really interesting to sort of pursue those. But I don't think we're able to, at least from this data, we're not really able to say much more about it.
It's a good reward for the brain.
Yeah, no. I mean, there's a reason for it, but calorically, but I guess in some ways it's interesting because you're right, sugar is good for the brain, but calorically that pellet is better if you're hungry. And what's interesting is that even though sugar's got this such kind of potent drive to the animal, which presumably why they go for it, dopamine seems to be able to know that actually the food is better at some level, for some element for this hungry animal. It's valued higher if we read the dopamine as a value response.
And,
Yes?
and did they use any antagonists or antagonists to find the mechanism of this difference?
No, I mean there's lots and lots of people that are working on dopamine circuits, food and different elements and probing the system really carefully and elegantly. We were going after a different thing and accidentally kind of found these effects essentially. And for some of you who know a little bit about animal behavior, you might also think about the fact that if you ask a behavioral ecologist to look at this kind of model, he'll say, well, that's perfectly fine, but it's almost certainly not what an animal's doing. There aren't many sort of rodent or monkey supermarkets where you're able to suddenly get nice, your 3 choices of food to choose between. That's not how the environment works. And instead, there's a class of foraging models that suggest that essentially what you have is a default starting policy of some sort. So this might be monkey go for those things. And you go along doing it, and then at some point you wonder, should I go and do an alternative, or should I continue with this? And although there's a lot of similarities with this version of decision making, you can see that the process and the fractionation points are quite different. And as of yet, we've got no evidence about whether or not this is closer to being important for the dopamine system than the other, but my strong hunch, and we're about to test some of these, is that this is going to be much more informative, these types of tasks, for able to unpick aspects of dopamine. And I guess for the last bit, what I'm going to do is describe some work which you've probably already been primed on. This is work that Laura was involved with, and also, to some extent, Oliver sort of coming off the bat for the pharmacology. And it's work where we've started to look at, instead of action selection as actions between options, we're looking at action initiation. So there's decisions about whether or not to make or withhold actions for reward. And obviously this is not, we haven't plucked this from nowhere. This is something which has been in the literature and people are talking about it. So this is from, I don't know if Emra's here yet, but that Emra, Mark Grita-Mazip, Ray Dolan and Peter Dan have been talking a lot about the fact that actually that there seems to be these interesting interactions between not just what you expect to get, but what you have to do, whether it's approaching or avoiding in different situations. And actually also in the voltammetry field, pretty much any study that's actively involved an instrumental response has seen something which looks like activity that's particularly driven by the action. So this was a cued action task that Mitch Reutemann ran, again, a long time ago. And he found that most of the time the animals responded quickly. You can't see that here. So most of the time there's a dotted line here that says, I think it's 0.1 to 133 seconds. So that's basically from cue to response. You know, there's a big variety of responses. And what he found is that basically when you time lock to the action, to the point where they press the lever, you see this ramping up to the lever press. And actually the peak response there is higher than if you just time lock things to the cue. So even though the cue predicts reward, it seems to be closer to the action you need to do in order to gain the reward than the cue itself. But again, this task is a bit like, this result sort of falls out of a reanalysis of the data, and so it doesn't really sort of allow you to probe this type of thing specifically. So this is something that we ended up going after. So there's an older picture of Laura, and Emily Syed did a lot of this. And again, this is another good example of actually work where we started off. This was meant to be experiment 1A to go in a different direction. And then we found it was so interesting that we never got beyond 1A. So this was meant to be a whole thing going towards dopamine and beta and Parkinson's disease and stuff. And this was just a task validation, essentially. And what we did here is, again, we've got a rat, we've got an operant box, we've got two levers, but we're going to make some important changes. So here, now, a rat initiates a response by making a nose poke, and then a certain amount of time later, you get a cue. And the cue might say, go and press a lever. So one cue says go and press the right lever, one says go and press the left lever, and if they do that correctly, they'll get a reward. But we also have a class of cues that says stay put. So basically withhold any response going to press the levers, either lever here or going to the reward magazine, which is on the back. And if you do that, only if and only if you stay in this vicinity of this nose poke port, will you get reward. And what you can imagine here is formally at least, that the levels of reward prediction at the time of the cue ought to be very similar in these cases, but obviously with parametrically controlling the action requirements. So just to formalize this, in the first experiment, we just had these options. So you had to go left, no go, and go right. Here you had to stay for a while in the nose poke. We made them do 2 lever presses for no good reason other than that basically extended this bit of time so that then reward delivery would happen at pretty much the same time if you went through one of the go sequences compared to a no-go sequence. And when we asked Rafael Bogac just to check whether actually this task would allow us to look at prediction errors, he did what was called an actor-critic simulation, where you can just very, very simple, so this is very chunky, like half-second blocks of data. We just said, what does prediction errors look like in this state? And the important thing is when the cue comes on, in all 3 of the conditions, you get this big reward prediction error-like signal there that's not significantly different between the 3 conditions. So in principle, theoretically, we ought to see a big dopamine response at the cue at this task. Okay. Here's a go trial. So again, the joy of voltammetry is you take your good animals and you look at individual trials. So here we've got, it's quite busy down here, so the head entry is the first bit here. The cue comes on this thick dotted line. The red is where the animal leaves the nose poke, goes to make lever press 1, lever press 2 leads to reward, turns round, goes to the back of the box and collect the reward. And there's a few things here. So one kind of looks like you'd expect, nice kind of response for a rewarded cue response. It's kind of extended. And actually, again, this is Josh Burke's seen this, other people have seen this, that there's something about the accumbens dopamine signal that when an animal is doing an extended set of things, particularly with different space, there seems to be this signal sort of extends over time, and it peaks just before magazine entry. So this isn't a reward response. This is just before the animal collects the reward, basically. So that's, I think, is quite an intriguing thing. That's the average across animals, go left and go right. So you can see that's one trial, but you can see that that's a pretty similar response across all animals. Just a quick thing, so we want to say, well, what are the parameters which we can see in this signal? So is it how rapidly the animal is moving? Is that what drives the dopamine release? Or is it something to do with reward expectation? So it doesn't seem to be vigor. So if you take fast response trials and slow response trials, pretty much overlap. If anything, the fast ones drop off earlier than the slow ones. So no sense that the vigor of the response is being read out in what we're recording in this accumbens dopamine signal. But if you split them in terms of most of the animals were slightly better at one of the two go options, go right versus go left, and actually that is a good predictor of the dopamine. So they're the one which they're better at, they're better performance at, you get a bigger signal and a more sort of faster signal, whereas the less good one is often slower in some way. So again, it sounds quite like the classic picture. Dopamine's released shaped by reward prediction and not vigor. And then obviously the key thing is what happens in that case where the animal has to withhold a response in order to get the reward. And this is a good example. So here, it's the head entry. That reward, the cue comes on, nothing. Reward's delivered, nothing. Nose poke exit, boom, there's your dopamine. And again, it doesn't seem to be something that's an artifact. This is across all the animals. You can see at the time of the cue, there's basically nothing happening until the movement. And again, you time lock to the response. So this is we reshuffle the data for when the action is made. And you can see that that seems to be the point when the dopamine goes up. Yes?
The time in these experiments is quite stable. I mean, it's 6 seconds and 7 seconds, right? So I just wonder about that, whether, the response is to reward mechanism related to
Say that again. Response to remote.
The time, the experiment, the conditions is under for all the experiments.
I mean, well, no, that's just I've taken 6 seconds of data. So that's not that there was a, that just sort of roughly covers pre-trial and a trial.
The Q and the reward is not.
Q is that, so head entry is the first line, Q is the second line, rewards somewhere over here. I mean, so for this one, rewards about there, for these one, rewards about there. And then it drops off after that. The ITI is about 5 seconds, but it's self-paced after that, so they can...
I just wonder about whether is there any time conditioning rather than the Q, the response to Q?
Doesn't seem to be. No, I don't think we're seeing anything like that. And so you can see there's a sort of little bit when they make the head entry and the sort of pause there, but we don't... I think there's enough variance in the animal's behavior. So if an animal performed perfectly, we should see no prediction errors because they should know everything. But there's enough variance in the animal's behavior, particularly actually that they will go back after the ITI and make a head entry response, which then doesn't start the trial. And so when we actually look, there's enough of those. So Raphael built that into the model, and that's why you see a prediction error at the time of the cue. Whereas if everything was perfectly stable and the animal had perfect knowledge, you're absolutely right that we'd see no prediction errors. Okay, so that's exciting. So dopamine release seems to, in response to report predicting cue, seems to be contingent to some extent on action initiation. So is it all action? Have we got, is it just that it's action because, it looks like reward very often because actually that's so often tied to actions? Have we been sort of going down the wrong path? So no, it's not, but I'll show you some nice examples why it's not. So here's an error. So the animal fails to stay on a no-go response, so he makes an action and then gets a cue telling him he got it wrong. And here you see a nice negative response when they get an error. So here's time lock to the cue, here's time lock to the action. So certainly not an increase at that point. But again, you might say, well, that could be overriding it. You basically, your prediction error that you've got it wrong could be driving that down, and there isn't time to see an action response. So this, I think, argues against that. So there's a small but significant class of trials where the animal gets a cue telling him to go in one direction, and for whatever reason goes and presses the wrong lever. So here, the cue's come on, the animal's correctly made an exit from the nose poke, has initiated an action, but has gone to do the wrong thing. And they only get told they're wrong by the feedback when they actually press the lever. So there's all the time when the animal's moving around the box, until they've pressed the lever, there's nothing explicitly cueing them they've got it wrong. And clever old dopamine knows this. So this is a standard trial, the cue response to the dopamine and the response. This is one of those wrong go trials where the animal is making actions, initiating an action and moving towards the lever. No rise in dopamine, no change in dopamine, and then this is the point where they actually get told in the experiment that they got it wrong. So somehow we've got clever, clairvoyant dopamine that knows that the animal's heading in the wrong direction and won't get reward for this trial.
Is there any method for omission?
No, we haven't. Yeah, we haven't. We didn't look at that. I mean, generally in most experiments, emissions, there isn't much that happens. And I guess that sort of feeds into, I mean, there isn't much change in dopamine. And I guess that partly sort of feeds into what we think about dopamine, in that it's providing some sort of threshold motivation for this degree to initiate action, essentially. Yes?
And how much time is actually between the release and those code and are those wrong option? Can they actually change and realize they're wrong?
Yeah, I think in principle they could. Yeah, so we don't know. So I mean, we do video them, but we've not been systematically going through the data to track them, and it's something we're going to certainly start to do, because I think that's an interesting question. And so I suspect that there's certainly so again, I took the slide out. So there's a really interesting set of trials where they do what's called a delayed go. And the delayed go is they kind of mix and match, that they kind of stay in the nose poke for a while, and then they come out and do a go response. So they're sort of like doing all the trial types at once. And that looks exactly like a kind of no-go response, that basically you see flat response when they're still in the food magazine, and then you see the go response come out when they come out and respond. So yeah, it's possible that within this, there's a few trials where they've gone off in the wrong direction and come back, and I guess you'd imagine that there may be some dynamics that have come in there, but we can't easily pick them out, I think, at the moment. Yes?
So at the point of cue onset, I guess, as measured by terminal release, dopamine doesn't predict whether the animal's going to get it wrong, but you can see that later. Right?
Well, it depends
at
on
the point of
yeah, I mean,
related to the cue on offset, cue onset at the left-hand side.
yes.
There seems to be no distinction, but that arises later.
I'm not sure. I mean, so basically, I guess 1 second. So dopamine with voltammetry, there's obviously diffusion to the electrode, and there's a time constant of about 150 milliseconds on the electrode. So applying dopamine directly to your electrode, you won't see anything for at least sort of 150 milliseconds. When there's a diffusion as well, it's going to be, it seems to be on fast electrodes, it's more like half a second before you're seeing things peak. So these separate pretty damn quickly. So you're right that it takes a little bit of time, but it's possibly not until the animal's set off, but it's pretty fast, because look here, by the time the animal's actually moved, which is half a second later or so, you're already seeing that.
With this, you would actually see the distinction earlier on at that exact point where you have the dotted line.
Do you mean here?
On the left-hand side.
In here?
Yeah.
Yeah, no, that's a good point. We were wondering if there would be anything in the pre-cue period that might predict them being more distracted or something. We never found anything. We've never seen anything. But I think it's a good idea. It seems plausible, at least.
So, isn't delay, as it refers to assess decision making, and is that related to response?
So in some ways, so I guess part of bringing up about the foraging type design is, in fact, we've sort of moved away in this from decision making, because we're telling them on every trial there's a right thing to do. So a kind of easy distinction about decision making is a decision making task is one where you're going to get something whatever you do, whereas this one is one where we've imposed rules on them and said when QA is on, you have to do this to get reward, and when QB is on, you have to do this to get reward. But I think that what I'm trying to lead towards is that there may be a link between, if we take this type of situation to a decision-making foraging task, which has a lot of these similarities of whether to initiate an action or not, then we'll see similar patterns of dopamine. only, but there's a right answer. So basically, when QA is on, if you don't press the left lever, you won't get reward. So there's a choice in the sense that every behavior is a choice between doing it and doing something else. But the value functions are one thing is rewarded and nothing else should be in this situation. And they know that. They've been trained for weeks and months. Yeah?
I have a question related to the measurement scale. So we are now on the molecular level. Would you find changes also on a higher level, like using fMRI scale? Do you think that?
Well, yeah. So that's where the Marc Guitard-Masip stuff comes in. He was looking at fMRI and doing pharmacology with fMRI and these kind of things. And he's one of the first people to say, actually, if you directly look at reward or reward plus approach action, really seems to be you need that approach action as well in order to get particularly sized ventral striatal signals.
So I think we've got to wind up in 5 minutes or
Yep.
so for the next session.
Yep.
Perhaps you can just interpret what you think this actually means.
So I will, let me see if I can flip to, so I guess we've linked it to prediction errors. So and this is all Laura's stuff, blah, 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 Oliver's. There we go. Yes. You've seen that once, that's fine. So basically, I think that, so what I'd like to do with this, and we're trying to write a review at the moment to sort of try and sum this up, is I think it's intriguing that dopamine release in the accord accumbens does take on a lot of the parameters which the classic Wolfram-Schultz prediction error-like story comes out. And we see that, and we see it repeatedly, and we see it in lots of cases. And I went in sort of quietly hoping to find no relationship and sort of go after it like that. And it's not. There's lots and lots of situations where we see very close relationship between what would be predicted by a prediction error model and what we see in the core of the accumbens. But you can't get away from the fact that for God knows how many years that the activation element of the accumbens has been there. And so what I'm hoping to do is that this provides some starting point to reconcile those two positions. That if we think of prediction errors as the signal of opportunities that are available in the environment, it might provide that activation and simple conditioned approach response to be able to go and investigate those things. And equally that negative prediction errors might not only pause behavior but even unlock alternatives. And so I think that this may provide a way in which we can start to bring those together. And I guess what I'd like to do is to sort of go, I think dopamine doesn't play a clear role in these sort of learned choice scenarios where there's economic options, the kind of things which I don't really think animals do, so maybe that's not surprising. But I do think that dopamine plays a really critical role in determining if a policy that may have been selected by cortex is carried through, that there's the motivation to do it, and possibly when as well. I mean, when I've talked to people who are outside of the reward-guided decision-making field about decision-making stuff, they say that when to do stuff is arguably more important than what you do in lots of cases. And the sort of ongoing hypothesis is therefore if we start to go back and think about these kind of sequential foraging choices, when we decide whether or not to engage and when we switch to an alternative, we might actually start to bring the relationship between those dopamine signals that we're recording and behavior really much more closely aligned than we are at the moment. And that's lots and lots of people have been involved at various points. So thank you very much.
I think we'll allow you just one quick question, but we really must get on to the...
So in the beginning, you found out you were a little doubtful about the translatability of this species. So what's your sort of general opinion of the translatability of what you showed?
Well, I think I didn't mean to sound doubtful. I think you should just be cautious. So I think, but what's intriguing is, so for instance, I mean, I was talking last week to one of the fly experts, and they were sort of looking at the way in which aversive things might activate populations of fly dopamine neurons. And what does a fly do in an aversive thing? He flies off. So a really critical thing. So again, in fear conditioning experiments, you get very different patterns. Whether or not you get hippocampal or amygdala effects depends on whether you run it in a box where they can only freeze and can't escape, or you do it in a Y maze where they've got options to move around. So I think that actually there might be a lot of commonalities in what dopamine is doing across species, and that's going to be really important and provides that link. But we just have to be really sensitive to what the species is doing.